Today in class 12 we will start with the incomplete dominance. So when experiment actually on pea plant actually we repeated using the order of other trait, it found that sometime the F1 had a phenotype that did not resemble either of the two parent and was in between the two. The ingredients of flower in case of dog flower, that is snapdragon or anthurium species, a good example of understanding incomplete dominance, because in case of this cross red flower, that is capital R, true breeding with white flowered plant, smaller, and when selfing that will F1 hybrid, that is capital R smaller with pink color, that is self pollinated. Again the result will be in the ratio of red is to pink is to white, that is to 1:2:1, that is genotype and phenotype ratio both the same. So here actually genotype ratio were exactly we expect in the Mendelian monohybrid cross, but as phenotype is also ratio had changed from 3:1 dominant recessive ratio. What happened in this R was not completely dominant over R, and this made responsible for distinguish the R as a pink, that is red and white. Explanation of this concept, if we see that is some alleles dominant and some recessive. To tackle this question we must understand the gene. Every gene you know that by now contains the information express particular trait in a diploid organism. There are two copies of each gene as a pair of allele. Now these two allele need not always be identical and heterozygotic. One of them may be different due to some changes. One is undergone further and which modifies the information that particular allele contains. So here also gene contains the information of producing an enzyme. There are two copies of gene, two allelic forms. So here also that if we see that the allelic gene transformation of substrate S, theoretically the modified allele could be responsible for production of the normal less efficient enzyme or the non-functional enzyme or no enzyme at all. In first case the modified allele is equivalent to unmodified allele and produce a phenotype that actually substrate S. So the equivalent allele pair common and functioning unmodified allele. The unmodified allele which represent the original phenotype is the dominant allele, and the modified allele is generally the recessive allele. So here the example above, the recessive trait is seen due to the non-functional enzyme or because of the no enzyme is produced. Now move on to the another example, that is co-dominance, that is discussing the F1 resembles either of the two parent was in incomplete dominance, but in case of co-dominance the F1 generation resemble both parent. A good example of this actually is red blood cell, that is determined ABO blood grouping system in human being. ABO blood group are controlled by the gene I, capital I. Plasma membrane of red blood cell is sugar polymer and protrude from the surface, and kind of sugar controlled by the gene capital I is three allele, IA, IB and small I. Allele IA, IB produces slightly different from the sugar, while allele small I doesn't produce any sugar. Because humans are diploid organism, each person possesses any two of the I gene allele, and IA, IB are completely dominant over small I. In other words, when IA and small I is present, that is IA expressed because I does not produce any sugar. When IB and small I is present, IB also expresses because together both express their own types. In this case it is called codominance. Here the red blood cell have both A and B type of sugar. Since there are three different allele, there are 6 different combination of these three allele possible, in total 6 different genotype of human ABO blood type. Here also you will see the diagram. So do you have here also realize the ABO blood grouping also provide a good example of multiple allele. Here also individual two allele can be present, multiple allele can be found only in population studies and made. Occasionally single gene product may produce more than one effect, like starch synthesis in pea seed is controlled by one gene. Two allele B and B, so capital B small b, starch is synthesized in BB homozygous, and large starch grain is also produced in contrast of BB homozygous small b. So after maturation of seed, capital B seeds are round and BB seeds are wrinkle. Heterozygous produce round seed, that is, and capital B seems to be dominant allele, but the starch gain produced intermediate BB, capital B small b seeds. So in starch gain size is considered the phenotype. Dominance is not autonomous feature of the gene product, that is information for it depends much of the gene product and production of the particular phenotype. There is in case of more than one phenotype is influenced by the same gene. So these are the actually called the codominant. So today up to that, next we will start with the another topic.